देख रहे हैं जेवी टीवी।
Dr. Sujit, good morning.
Good morning, sir. I can hear you.
Good morning, yes. So it is now time to start the webinar. The participant have come and the participant will be keep on coming. So it is a matter of great pleasure that a webinar is being conducted today on production of artificial seed from seedless plants. And it is being conducted with our organization with which we have one MOU, and it is a collaborative activity. And the name of the organization is Cytogene Research and Development. And Cytogene Research and Development is based in Lucknow, which is very much active in biotechnological based research. And it is headed by Dr. Sujit himself. Dr. Sujit has more than 10 years experience. Dr. Sujit has more than 10 year experience. And he has more than 60 publications, has conducted seminars and conferences and other technical events. And let me tell you for your information, Dr. Sujit, today our listeners are our student of BSc Agriculture, BSc Honours Agriculture, BTech Food and Biotechnology, BSc Food Science and Technology. And what I think, my thinking is that agriculture and food and biotechnology are very much correlated with each other, and they make a good integration of research. Because if agriculture is not there, what agriculture, biotechnology and food technology I will do? So with this now, over to you to start the webinar on this topic. And the student, you listen attentively, and if you have the questions during the question answer session, you can ask the questions. So over to you, Dr. Sujit. Thank you very much.
Thank you so much, sir. Really nice introduction by you. And I believe you people are so active while organizing all these lectures and for benefit of the students. Definitely, it is a time of pandemic situation, but I think everyone is getting used to it. And you have done, it's nice to be here. I think if we have to personally visit to university, it will be difficult at this time. But you have getting benefited to all these students by using all this technology. I will not take much time on this. I wish to a very good morning to everyone, and I believe, as sir already informed, like agriculture, food technology, and biotechnology resembles with each other. And I think it is now the time to make Atmanirbhar. We have to be integrated with each other. Okay, can you see the presentation?
Yes, sir.
Okay, good. Production of artificial seed from seedless plant. Name itself shows what actually we are going to do in this. Just if we'll go to history, who has started with this? That is Mercier in 1977, also named as manufacturing seed, manufactured seed, or maybe synthetic seed we can say. Sometimes in seed we call it. In this, we take an embryo, as I already told you, from tissue culture, and we encapsulate in encapsulating material like sodium alginate or something like. Why synthetic seed is needed? In some of the horticulture crops, seed propagation is not successful due to heterozygosity that actually creates problem when it is cross-pollinated. Sometimes सीड साइज बहुत छोटे होते हैं जिन्हें इजी नहीं होता है कल्टीवेट करना. प्रेजेंस ऑफ रिड्यूस्ड एंडोस्पर्म, sometimes it is reduced, sometimes it is damaged also, we can say, because of environmental conditions. So it is not easy. No seed are formed like banana seed, banana plant, and many more options you have. What actually the characteristics of synthetic seed is? High volume. The best part is large scale propagation method. Means for a normal natural seed, we need to give a season. Suppose if you want to complete some seed of like any plant, like wheat or rice, you have to go for by season. This year if you will grow, next year you will be getting the seed. But here in propagation, overnight you can start. Within a 15 days, you will be getting seed as much as. Like now, earlier what happens in COVID cases, if you compare, vaccine production year. But this time they have developed everything within a year. That means everything is getting fast because of the requirement. So if we have more requirement of seed, we cannot wait a year to grow it and then getting it for our agriculture. So it can be produced by artificial seed production, and it will give within a month you will be getting all the seeds. The best part that techniques we regularly use is tissue culture, but that has a limitation. Like whenever it will grow, you have to definitely the same time you have to go for micropropagation and further primary and secondary hardening. But in this case, that we can postpone the primary and secondary hardening. Maintain genetic uniformity. In environmental condition, there are many mutation and everything is done. Sometimes pest attacks is done. So because of that, genetic diversity mostly it is maintained, but the year it is getting down and pests are getting more harmful. And what we do, we use chemical based all these nutrients for plants. That is actually not good. So if we are going to maintain an organic compound, it will be better if we go for the micropropagation, because in the plant whatever the genetic component will be, it will be continued till 10th generation as well as 50th generation also. So throughout all generation the genetic uniformity will be all maintained. Next is direct delivery of propagules to the field, and this is the most important. Actually in tissue culture we cannot go directly to the field. If we have callus, then we have to go for media preparation like MS media, and we have to grow in that. Then primary hardening, then secondary hardening. Then we are going to the field. But in this case, we directly go to the field. Directly we use those culture callus, and we go through the field, and it gives a like better condition than growing tissue culture and all. Lowest cost, as I say already, it takes much less time. So definitely, if some plant is taking 1 year and this is taking 15 days to 30 days, so definitely in 1 year, 12 times you can do the same thing and within the lab. So that is why the cost will be definitely very low. All the spaces occupied are for a shorter duration of time, and multiple time you can do a production. So in that way, it will lower the cost. Rapid multiplication of plant, as I already said, within the lab you can do whole day, night, every time. So mostly you will be getting benefited. Based on technology established so far, two types of synthetic seed are known. Two means desiccated synthetic seed and hydrated synthetic seed. Desiccated means continuously you have to take a naked seed and encapsulate into this. And hydrated, what we do, we like cover with sodium alginate and all. Not much different, but here we remove the waters through desiccation. So the chances of like damaging a seed is less. If you can see here, somatic embryo either naked or encapsulated in this compound followed by their desiccation. So it will be compact and there is no chance of breakage of those seeds. While in hydrated seed, this is sensitive to desiccation, so we cannot like desiccate every seed. I hope you understand what is desiccation. That is just a removal of water from all the seeds, so it will become dry. Directly we cannot heat, so we go through desiccation. So in that case, if the seeds are sensitive, then we will go for directly to hydrated seed we will produce, and then we will encapsulate it. What actually the limitation is there for natural seed, which is already available throughout the year and near everywhere. Maintaining genetic variation are difficult. As I already said, many attackers, many pests, many mutation is continuously going on. So if we have to target some genetic variation, or we have manipulated some genetic, so we need to maintain it. So which is difficult in natural condition. Many fruit crops are difficult to produce, Yes, there are many fruits. Like in Simla and all these region, apple is growing very normally. But in lower region in Rajasthan, in UP, it will not grow. So that does because of environmental conditions. So in that case, what we can try, we can manipulate it and artificially we will produce the seed because we are going to introduce our genetic material in this going on the Kesar, and they are trying to produce in Nilgiri region, which is already available in Himalayan region, but it is not available in Nilgiri. So they are producing the Kesar seed. If they can improve on that, it will be really beneficial for them.
Yes, because Kesar is very costly, and if it is possible for that, it is.
Because it is very costly, if they will develop techniques for that, it will be beneficial to everyone. And which is already easily available at 10 rupees kg, it
Hmm.
definitely will not be a good business.
Yes.
Mm. Okay, I hope everyone is having some idea. So definitely you will have something new to go ahead.
Okay. So,
Please,
sir, in the last, you can suggest our students for if there any scope in the research or in the some production corporation for regarding this. You can suggest them.
there are actually, if you visit to Department of Horticulture, they have many programs. But from last this 1 year, because of COVID, everything has been stopped. I think again they will start from March. They have many plans for students also. They can visit, they can learn the technique, and then they can start. The Department of Horticulture is helping to the person who is interested in this all the things. So by the financial also they assist in that case. So if they have any idea, first they have to actually collect some ideas to work on this. Then they can produce their proposal to Department of Horticulture. They will definitely help.
Okay. So students can take some training type of from
There are
their.
some plants also, not only plants. Like they have many schemes. Because of this 6 to 8 months, every scheme has been hampered. But from February or March onwards, you can regularly visit to the Department of Horticulture. They put it on their website also. They have many trainings and many visits they are organizing just to improve your technical knowledge and getting some ideas for business purposes. So you can be contact with them. They will help definitely.
Okay, sir. Thank you. So, students, if you do not have any questions more, then we can say thanks to sir. Sir,
Yes,
it was really very good and informative for students, and definitely the students will be benefited from this information. Okay.
sure,
So,
sure.
okay,
Thank you
sir.
so much, sir.
Thank you,
Sure,
sir.
we'll do some better work later. It's just starting. We have actually mostly we do in molecular and all analysis. So some days later we will again try for that also.
Okay.
Thank you so much. It's been nice to introduce you all, and I hope you people are doing really great job. At least during this time, the student will be benefiting much, and I wish you all the very best for all this organization. Thank you so much, sir, for inviting us. Definitely, it is an honor for me. Thanks a
Thank
lot.
you.
Thank you.